Adderlings, in this video we are going to discuss about development of embryo in plants. Before discussing about this embryo, we will discuss how this embryo is formed, how this fertilization in flowers takes place. Firstly, the pollen grain, which is the male gamete, has the two layers, the outer exine layer and the inner intine layer. This exine layer, at some places there is a gap is present. This gap is known as the germ pore. And inside this pollen grains have two cells. One is large cell or tube cell and another spindle shaped vegetative cell. This pollen grain, using some external agents, it falls on the female gamete or the female reproductive organ, that is pistil. These external agents may be wind, water, air, etc., insects, birds, etc. falls on the female reproductive organ, that is pistil. This pistil has the three parts. One is stigma. Stigma is a bulged part, and below the stigma is a long slender like style, and below the style is the ovary. Inside this ovary, the ovule is present. Inside this ovule, the embryo sac is present. In embryo sac, towards the chalaza end, we will see three antipode cells, and at the middle, there is two polar nuclei. And towards the micropyle end, there is one large central cell known as the egg, and beside this large central egg, there is two cells known as the synergids. When these pollen grains come and falls on this stigma, using from this germ pore, the germ tube will be developed. This large cell or the central cell will gives rise to the germ tube. This germ tube will leads into the stigma and through the style it reaches the ovary. And the generative cell, which is in the pollen grains, undergoes one mitotic division and forms the two gametes, and these two gametes are the male gametes. These male gametes, along with this tube cell or the germ tube, come and reach to this egg. One male gamete fertilizes with the egg and forms the zygote, and this zygote leads into the embryo. And second male nuclei fuses with these two polar nuclei and forms the primary endosperm nuclei, which is the triploid. This primary endosperm nucleus gives rise to the endosperm. Now we will see the development of embryo. Now we will discuss how this embryo is formed in dicotyledon plants. After fertilization, this zygote undergoes mitotic division, and division is transfer division and forms the two cells. Out of these two cells, the cell which is towards the chalaza end is known as the terminal cell, and the cell towards the micropyle end is known as the basal cells. This terminal cells undergoes many divisions, and it divides in all planes and forms the four cells, and these four cells is known as the proembryo. And these basal cells undergoes many divisions, not a single division, many divisions, and all divisions are transfer divisions and forms a long structure known as the suspensor. And the cell of the suspensor towards the micropyle end is round and bigger, known as the astorial cell. This suspensor helps in pushing this proembryo inside to the endosperm. Imagine this is a zygote, and above this zygote is the endosperm is present. This suspensor helps to push this proembryo into this endosperm. This proembryo divides further and forms the 8 cells. Actually, this proembryo is a round structure. We cannot see all 8 cells together. 4 cells are present toward this side and 4 cells will present back side. To understand, I wrote like this. Keep on increasing this proembryo octant, the suspensor cells will suppress, and the suppressor cell which is attached to this proembryo will act as a hypophysis. Later this proembryo octate divides, but this time it divides periclinally. In periclinally we can see the two layers of cells, one is outer layer and inner layer. At the same time, this hypophysis undergoes division and form 8 cells. Out of these 8 cells, the 4 cells which is present towards the micropyle region will give rise to the future root cap and root tip of the embryo, and the 4 cells above or towards this chalaza end are attached to this proembryo octant will give rise to the root cortex. And in this proembryo octant, the outer cells are known as the dermatogen. This dermatogen will give rise to the epidermis, which is the outer layer of the future embryo. And in these inner cells, the half of the proembryo octet which is attached to this hypophysis will give rise to the hypocotyle, and above this half which is towards the thallus end will give rise to the plumule and cotyledons of the future embryo, and this suspensors and astoria cell will start degenerating. Now at this stage, this above half of the proembryo octet will give rise to the plumule and cotyledon, and this half will give rise to the hypocotyle, and of hypophysis the upper two which is towards the thallus end will give rise to the root cortex, and lower two which is towards the micropyle end or lower four cells will give rise to the root cap and root tip. And this stage this embryo is seen like a globular shaped, and later this undergoes many changes and leads to this. See, this proembryo undergoes many divisions and forms plumule is present here, and this becomes two cotyledon, one cotyledon and two cotyledon. This half of the proembryo octet will leads to the hypocotyle, and this hypophysis upper four cells will becomes root cortex and lower four will becomes the root cap and root tip. At this stage this embryo looks like a arc shaped embryo, and later this will become like this. See, two cotyledons are raised here, plumule is present here, and below this is the hypocotyle, and this is the root cortex and root tip and root cap. At this stage it looks like a horseshoe shaped embryo. Later the embryo is formed. In this embryo, this is the embryonical axis. At the center there is a cotyledonary node from which the two cotyledons are arises, and at the upper to this embryonical axis is the plumule which gives rise to the future shoot system, and lower side to this embryonical axis is the radicle which gives rise to the future root system, and above this radicle and between this cotyledonary node is the hypocotyle, and in between this cotyledonary node and plumule is the epicotyle.